Morning, Epidemics. Welcome to class. Just a couple of reminders before we get started today. There is a review quiz and a journal assignment that I posted on Friday last week that are due this Friday before midnight. The project is coming up due on April 30th, which is just a couple of weeks away now. But it's almost the end of the semester. Our last lecture is going to be on May 2nd. The semester technically goes for one more lecture after that, but I'm not going to lecture on May 4th. I'm going to give you time if you're behind on any assignments, if you need to make anything up. I'm going to give you some time off, so to speak, to catch up on any assignments. And it can be in my class, it can also be in other classes, depending on how stressed you've been all semester. So I also wanted to clarify one thing before we get started, that when I was grading recently, I noticed that there was some confusion about life expectancy. This is a very confusing term because when people who are not scientists use the phrase life expectancy, they think it means how long am I going to live? It doesn't. It means the average lifespan of a group. So you can have a group of people, say hunter-gatherers. You can look back and say hunter-gatherers had an average life expectancy of 35. Well, in scientific terms, that's accurate. That was the average lifespan of that group of people in that period of time. But what that doesn't tell you is that if you made it to adulthood, you weren't likely to make it to age 70. But hunter-gatherer societies back in the ancient past had a childhood mortality rate of around 50%. So half of the people did not make it to adulthood. And so that brings down the lifespan, the average lifespan or the life expectancy of that group. It has nothing to do with individual lifespans. It's talking about a group. Okay, so we've been talking about antibiotics. And I want to talk about a couple of problems that modern society is having related to antibiotics. So the first thing I want to say is how should you take antibiotics? Now, I'm not talking about with a full stomach, with an empty stomach, what time of the day. No, none of that stuff. If you have a virus, do not use antibiotics. It doesn't do any good. Antibiotics only work against bacteria. Do not take them as a preventative or prophylactic. So you don't want to take them because somebody in your house has a bacterial disease and you're afraid you're going to catch it. Please don't do that. Unless you are extremely high risk for an infection or a doctor prescribes them. So sometimes if you're going in for some certain types of surgery, they may put you on antibiotics as a precaution. But as a general rule, try not to do that. Never take them unless directed by a doctor. in most parts of the US, you can't just go to the drugstore and buy antibiotics. Now, you can do this in other countries, and you may know somebody who has antibiotics and didn't finish them, or you may have taken antibiotics in the past and have a few pills left in the bottle, saved them for later. Or if you're like me, I do a lot of traveling to less well-developed countries. And so travel doctors will often give me a prescription for antibiotics in case I get really severe digestive problems. We'll be delicate there. And I always take the antibiotics with me, but I never take them. I don't get sick, so I don't need them. So I come back, put them in the medicine cabinet. I don't take those unless I'm in a place where I don't have ready access to healthcare and I get very, very sick. That's the only time I would take those. And I wouldn't take them for like a head cold. It would literally be extreme stomach distress. Never use something that is antimicrobial that is not FDA approved. So there are a lot of things out there that are antimicrobial, and we can use those for things like cleaning. So for example, bleach is an antimicrobial. It's a great cleaning product. You really shouldn't drink it or inject it. I hate the idea that I have to say this, but people were literally drinking bleach in the early parts of the COVID epidemic and getting very, very sick from this. So that's not a good idea either. Now, why am I saying all of this? Why am I telling you all of these conditions not to take antibiotics in? And the reason for that is we are seeing something called antibiotic resistance. This means that there are bacteria out there which are no longer being affected by antibiotics. The antibiotics are not killing them. Resistance is a worldwide problem. And there are bacteria out there that are resistant to every single antibiotic we have. If you remember, we looked at the mortality rate before the development of antibiotics and how it changed with antibiotics. Things like certain infections would have a 25% mortality rate. And that got reduced with the first drugs down to 5%. And those weren't great drugs by today's standard. So we don't want to go back to that world. And so antibiotic resistance is a risk for us going backwards with the progress we've made with antibiotics. We estimate that there are over 1 million deaths yearly due to antibiotic-resistant bacteria. In the US, it's about 35,000 every year. So it's a big problem. This is a woman I know. I'm not going to tell you her real name. She is on Twitter, and she is an ecologist, and she was studying ground squirrels. And specifically, she was measuring the size of their den openings. They live in the ground. So she was measuring den size openings and various stuff. And a ground squirrel came out of the den and it bit her on a finger. This wound got infected very quickly. She was in the hospital within 24 hours. The infection was multiple drug-resistant Staph aureus, Staphylococcus aureus, or MRSA. So MRSA is one of the most dangerous ones. And you can see this little tiny bite wound. Here's the skull of a ground squirrel. Those teeth, those are big teeth. It's not a pleasant bite, but it ended up being a life-threatening situation. She spent over a week in the hospital. She had multiple surgeries. The infection went up her arm, and they had to do multiple surgeries. You can see this scar where they had to open up her arm and literally remove infected tissues. And eventually, she had to have that finger amputated. So her second finger is only about that long. She lost the last bit of it. This is what antibiotic resistance can do in a young, healthy person.
I had a similar situation where I was hospitalized for a week with a bone infection in my left middle finger.
Oh, goodness.
Yeah,
that's not good.
Luckily I still have all 10, but I didn't know it was infected for quite a while.
Mm-hmm.
I treated it like it was just a broken finger because it felt like a broken finger. I cleaned out the wound and I essentially just splinted it. I suppose I kind of made a tourniquet without realizing because the infection didn't really spread. But I was on I was going to mention this last class. I was on vancomycin for a while through a PICC line for roughly a month.
Wow, that's tough. Thanks for sharing. For those of you who don't know, vancomycin is one of the antibiotics that's kind of reserved for multiple drug-resistant infections. So you're not getting that unless you really, really need it.
Yeah, and we actually had to stop it early because I began to develop a bit of a rash as a result of it. And getting the PICC line taken out was not nearly as unpleasant as I was anticipating.
Well, that's good.
Sorry, what?
No, go ahead.
One more thing.
Yeah.
They actually had to lance and drain my finger with a scalpel without any sort of numbing or like I forget the term, a nerve block, something like that.
Right.
Nothing
so no
at all.
Painkillers or anything.
Yep. Fully conscious, fully aware the entire time.
Ouch.
That doctor was not gentle.
Oof. I bet that hurt.
Probably the worst pain I've ever experienced so far. Hopefully ever.
Let's hope so. I can't believe they didn't give you a local anesthetic there.
He gave me the option. He explains it as either you'll have a terrible searing pain in your hand for like 5 minutes before it goes numb, or you'll be in the worst pain of your life for like 30 seconds. It was a tough call.
Yeah.
And it didn't help that he mentioned how long the needle would have been too.
Oh. Yeah. I've had an injection in a joint, which I'm assuming was where they would have to do it for that. That was absolutely some pretty nasty pain. So I can imagine. I think I still would have gone for it.
Yeah, maybe. He did mention it would have gone in between the webbing of my middle finger on either side.
Oh yeah, that's going to be a rough spot. Ow. Oof. Well, I hope you don't ever have to deal with that again. I hope nobody has to deal with that again. Oh,
Me
man.
too.
Okay. One of the difficulties with drug resistance is once a bacteria, any bacteria, develops resistance to an antibiotic, there is going to be resistance to that antibiotic from then on, basically. You can't set the clock back and get rid of all bacteria that have resistance to a specific medication. It unfortunately does not work that way. And I want to give you a quick quote by Alexander Fleming. Remember, he was very involved in the early development of antibiotics. And he was awarded the Nobel Prize in 1945 for the discovery of penicillin. And in his acceptance speech, he said, "The greatest possibility of evil in self-medication is the use of too small doses so that instead of clearing up infection, the microbes are educated to resist penicillin, and a host of penicillin-resistant organisms is bred out, which can be passed to other individuals and from them to others until they reach someone who gets a septicemia or a pneumonia, which penicillin cannot save." And he was right. He predicted that there would be resistance, and there certainly is. So how does resistance come about, and how does it spread? Basically, when we talk about bacteria, they divide very, very rapidly, and they tend to occur in relatively large numbers. So a bacterial infection is going to be thousands or millions of bacteria. And they're all dividing. And like I said, they can divide very, very quickly. In as little as 30 minutes, the population can double, which basically means each bacteria divides and produces 2 offspring in half an hour. A lot of them are slower than that. That's one of the faster ones. So because they divide so quickly, there is a higher mutation rate, which means that their genes can change relatively quickly. And this is a form of evolution. And if you've got a bunch of bacteria, you have one variety that has some resistance to an antibiotic just because of random genetic diversity. You take an antibiotic, it destroys or prevents these bacteria from dividing. Most of the normal bacteria will either not be able to divide or will die. The resistant bacteria then become more common. They continue to multiply. The antibiotic is still there. The vulnerable bacterium can divide a little bit, but they keep getting killed off. It doesn't really last very long. And eventually, the entire infection is resistance. So, one of the things to note about resistant bacterium is that very often what allows some bacteria to be resistant also negatively impacts their ability to divide, to survive, or to compete with the vulnerable bacterium. So the vulnerable bacterium in normal situations are healthy and outcompete the resistant bacterium. It's only when the normal bacteria get killed off that that resistant strain starts to multiply. So what kinds of things do we see? We see that mutations that bring about resistance happen about 1 out of every 10 million cells. Well, one cell dividing rapidly can reach 10 million, oops, in about 6 hours. So this is going to happen really, really fast. So what kinds of things happen in the bacteria that allow it to be resistant? One of the things is, if the antibiotic goes after a specific enzyme, and this can be an enzyme is a protein which does something in the cell. Enzymes basically do everything in the cell. If it needs to be done in the cell, there's an enzyme that does it. And so antibiotics will target certain enzymes, and the bacteria can evolve a change in the genes that results in a change in the structure or shape of an enzyme. So a mutation arises. The enzyme usually is not improved by mutations. But in this circumstance, because there's antibiotics around, having a different shaped enzyme means the antibiotic cannot target it. Whenever you change the shape of a protein in a cell, antibiotics may not recognize it anymore. Our immune system may not recognize it anymore. So shape change is very, very important. It's how come we keep having waves of COVID, because COVID changes the proteins on the surface that our immune system recognizes. And so our immune system just doesn't recognize the different shaped protein, and we can get reinfected. And if it spreads, we get a new wave. Another thing can be changes to the cell wall or cell membrane. These can reduce what's called permeability. Permeability means the ability of an antibiotic to get inside the cell. So literally that barrier on the surface changes so that the antibiotic can't get in anymore. If the antibiotic can't get in the cell, it's not going to do anything. One of the other things that can happen, there are pumps in cell membranes. And these pumps can be, in normal situations, they're used to export waste material and import certain nutrients and various things like ions or water or all kinds of stuff. It facilitates or helps the movement of materials across that membrane. And these pumps can be modified to export antibiotics. Now, these pumps require a lot of energy. So in scientific terms, we call these expensive. Energy is basically, if you use too much energy, if your cell uses too much energy, it cannot continue to function. So there is a cost to the cell for having these pumps if you don't need them. But if there's antibiotics around, even if the cell is not doing as well because it's spending all of its energy on these pumps, it's able to outcompete the other bacteria. Okay, so those are some of the ways that bacterial antibiotic resistance shows up in bacteria. And I want to show you a video that I forgot to pull up. So I'm gonna see if I can find it. Okay, here's the video. Can you guys, do you guys see the screen that's going to YouTube right now?
Yeah.
Yep, I see it.
Okay, good. So what we're going to see on this video, this is a giant growth plate. It's huge. And this growth plate has different layers associated with it. So
much, barely more than E. coli can survive inside.
So what's going on here is on the outer layers, there's no antibiotics. Then there's a little, and more as we move to the center.
Where does the poop go?
What the hell?
If someone's backed up and constipated for 2, 3 days, or even a week.
To understand how quickly bacteria become resistant to even the most powerful drugs, we visited Harvard or Cipro, a broad spectrum.
Okay, hang on. There we go.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. At the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, of course, some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. Then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
What did you guys think? 11 days, and that strain of bacteria became resistant to 1,000 times the lethal dose of antibiotics.
That's insane.
It is insane.
I found it interesting that it like stopped after the like 10 times or whatever was it. Yeah, after 10 times it like stopped and then it started mutating again, and then like after that it just kept mutating and like slowly getting through, but it stopped after that first wave.
Yeah, it kind of had to pause each time the antibiotic dose went up. Yep. Okay, so bacteria normally divide by what's called binary fission. So basically, they copy their DNA, they make extra proteins, they make extra cell walls and cell membranes, and then they divide into two. Now, this is the normal way bacteria divide. But there are several ways that bacteria, so we call this binary fission, we call this asexual reproduction. So there's no sex involved. There's only one individual at the start and two individuals at the end. There's no exchange of genetic material. Sexual reproduction happens when you've got genetic material from one individual, genetic material from another individual, and they combine or exchange some of that genetic material to create a genetically unique third individual. And there are ways that bacteria are able to get new genes. There's conjugation, transformation, transduction, and we're going to talk about each one of these in a little bit of, well, not a lot of detail, but a little bit. I'm going to explain what they all are. And these are all ways that bacteria can get genes from each other. And so these genes may or may not these genes can be involved in any kind of process in the cell, and they can go between different species of bacteria. So a bacteria that's called E. coli can give antibiotic resistance to something like Staph aureus, which we were talking about as MRSA. And so you can get these swaps between very, very different strains. This would be as if you could trade genes with a bird. And instead of having hair on your head, you could have feathers. We can't do that. Animals cannot do that, but bacteria can. So we're going to start with conjugation. So conjugation is very often described as bacteria sex. What happens? We have two bacteria. Again, they don't have to be the same species, just have to be bacteria. This bacteria, most of the DNA is in one large circle, but they can have these smaller circles of genetic material called plasmids. Plasmids are usually the part that is transported from one bacteria to another. So this bacteria has a plasmid that allows it to grow this structure that forms a bridge between the first or donor bacteria and a recipient bacteria. So this bridge is kind of like a hollow tube between the bacteria. The donor will make a copy of that plasmid and send it over to the recipient. So now the donor and the recipient both have the same plasmid. Plasmids often contain genes like ability to digest a specific food. So you can share the ability to eat something that the bacteria doesn't normally eat. These can also be virulence factors. We've talked about virulence factors before. These are things that allow the bacteria to be able to infect organisms more successfully. They can also be antibiotic resistance. Now, plasmids, a single bacteria, first off, if it has a plasmid, it usually has multiple copies of that plasmid. And it can have multiple different plasmids. But plasmids are, it adds a layer of complexity to asexual reproduction. So if you have a bacteria with plasmids and it does asexual binary fission, the daughter bacteria may end up with different amounts of that plasmid, or one of them may not have the plasmid at all. So a strain of bacteria can very easily lose the plasmid and produce offspring that don't have it. So that can happen normally. They can gain, they can lose these plasmids over time. We can also talk about what's called transformation. So transformation is a situation where a bacteria dies. It can die because you put bleach on the kitchen countertops. It can die because it couldn't get enough food. It can die for any number of reasons. And that dead bacteria, its chromosome, its DNA will fragment. But here comes another bacteria. And this other bacteria is literally crawling over the surface where the dead bacteria is sitting, and it can pick up DNA from the dead bacteria. This is really kind of freaky to think about. I'm not going to give you the analogy that's in my head because it's really gross, but you can kind of think about it yourself, getting bacteria from something that's dead. So it can pick up the bacteria and it can add that bacteria to its main chromosome, or it can have it as a plasmid. So that's another way to get new genetic material. And this is actually both of these forms, conjugation, transformation, and as we're going to see, transduction. It's changing the genetics of a pre-existing organism. We can't do that. Animals cannot change our genetic material. At least we can't do it deliberately, and we can't pick up new traits. You can damage your DNA by doing things like smoking or suntanning. That's not what I'm talking about here. So this is not something that we can do. So it's really bizarre. Again, I want to remind you guys, let me annotate this. This is not a good website. They have lots of bad information, even though it tends to come up frequently. They make good graphics, but I found stuff on there that's not, a lot of stuff on there that's not right. All right, so the last one, transduction. Okay, so this is, I keep saying this is freaky. This is another freaky thing. We have a bacteria here. And on the surface of the bacteria is what's called a bacteriophage. Bacteriophage is a fancy word for a virus that infects bacteria. That's all it means. It's a virus that infects bacteria. So this bacteriophage has genetic material inside of it. This is just kind of like what happens when you get infected with the virus. the virus sends its DNA into a host cell, infecting that host cell. That host cell then starts to make more viruses. Same thing your cells do when they're infected. The virus turns the infected cell into a factory for making more virus. Now, viruses produce millions of individual viruses from an individual cell. And some of those are going to pick up some of the host cell DNA. So you can see there's a little bit of fragment. If you're not colorblind, that's the downside to this one. This little bit of blue here is the host bacteria DNA. These guys are just viral DNA over here. So those viruses come out of the cell and they infect another bacteria. Only this time, this virus has some DNA from our virus in number 1. So it spits that viral DNA, the little curve here, as well as the bacterial DNA from the other bacteria into the host cell. And what happens, there's not enough viral DNA for this cell to make more viruses, but it can take that bacterial DNA and either turn it into a plasmid or put it in its major circular chromosome here. So it can take up bacterial DNA that was transported between different bacteria by way of a virus. And so now it has new genes. Biology is just absolutely freaky. You don't even know the half of it. I don't know the half of it. And I'm constantly in awe over how things work in biology. Okay, so let's look at the history of resistance. So a lot of antibiotics are naturally produced by microbes. Fungi produce a lot of antibiotics. Bacteria can produce antibiotics against other bacteria. So this is important in non-infectious situations, especially bacteria against bacteria, to reduce competition for resources. But it can also be a defense against infection. So that other bacteria may normally be able to infect a fungus or another bacteria. They can infect each other. Or it may kill and eat the other bacteria or the fungus. And so these naturally occurring antibiotics are to fight off that. In the wild, the amount of antibiotic produced is usually very, very low. So it's more likely to discourage bacteria rather than treat an infection. There's, if the infection has already started in, say, a mushroom, it doesn't really have enough antibiotics naturally produced by it to fight off that infection in normal situations. It can ramp things up and there are things that it can do, but it doesn't naturally produce those high levels. So we discovered these antibiotics and had to develop methods to extract them. So how do we get them out of the fungus or bacteria that make them? How do we purify that so that we have just the antibiotic? Or how do we make copies of that antibiotic? How do we manufacture that antibiotic ourselves? And how do we get a high enough dose to be useful? If you think about it, if you're a fungus and you're the size of one of these little discs here, the amount of antibiotic you need to fight off an infection or prevent an infection is a lot less than what you're going to need if you're a human-sized organism. So we have to ramp up how much gets produced. In the United States, by the 1950s, penicillin was available. And how am I misspelling penicillin? That's what I'm doing. Was available over the counter or OTC. So you could walk into the drugstore and get penicillin, no problem. It was in everything. We still see this as a problem today. These are products that I found on, I think it was like a Staples website or something like that. Here is a heavy duty stapler that has antimicrobial in it. Here are scissors that have antimicrobial agents in it. Here are file folders with antimicrobial agents in it. Why do we need file folders that can kill off bacteria? We don't. And really, this is just a very bad use of microbial agents. Now, some things are naturally antimicrobial. If we're talking about the scissors, a lot of metals are antimicrobial. So bacteria cannot survive on them. So if you've got metal doorknobs, especially if they have copper in them, anything with copper in it is going to be antimicrobial. It's not going to get through your skin into your body if you're wearing copper jewelry, for example. But, you know, if we're talking about doorknobs with copper in them, you're not going to get microbes growing on it. You're not getting bacteria surviving on that. In the 1950s, penicillin was promoted to encourage growth in children. So the idea was children taking penicillin would grow larger and faster than children not taking penicillin. So how much is this a problem? Well, it depends on what they mean when they say antimicrobial. If they mean they've got antibiotics in it, it's a big problem. But if it means that the surface is not likely to grow bacteria, like metal surfaces, it's not really a big problem. So antimicrobial as a word doesn't really mean much. It could be antibiotic, which is a drug, and that is a problem. But it could also be soap and water. Soap and water are antimicrobial. That's why we wash our hands. Vinegar. Vinegar is good antimicrobial. It's less harsh on surfaces than bleach. Bleach is very antimicrobial. It'll damage surfaces a little bit more. Soap and water and vinegar, excellent cleaning agents. You can clean a lot of surfaces with just those products. And these are not going to be a problem. Because the way all of these work, it's not something that's going to give rise to resistance. Soap and water is antimicrobial because it's literally carrying the bacteria off of your skin. Vinegar and bleach both have kind of extreme pHs, and it's very, very hard, if not impossible, for the types of bacteria that can infect us to also be able to survive in those extreme pHs. There are bacteria that can survive in extreme pHs, but the pH in your body is not that extreme. It's not going to be those same levels of pH. So if the bacteria has survived or has been able to evolve to withstand the extreme pH of bleach, it's not going to be able to survive the less extreme pH of your body. So we have antibiotic resistance. It's becoming a problem. And there's a lot of reasons for this. We're going to start with overuse in medicine. Not just talking about the 1950s when everybody was taking it. When antibiotics were first discovered, they were considered miracle cures. And they kind of were. They were able to cure diseases that had previously had relatively high mortality. It really brought especially childhood mortality, but also mortality in older people. It brought both of those down significantly, which caused a huge spike in life expectancy for the population. So doctors started using them. And sometimes it makes sense. Sometimes you need to use them to treat infections or prophylactically, especially if somebody's going into surgery. Here's the thing. Doctors work on patients and they work on individuals. So my doctor, I go see my doctor. She decides that I go there because I'm sick, and she decides I need an antibiotic. She's treating me. And even if whatever is wrong with me is bacterial and is not likely to progress, or she thinks it might be bacterial, but she's not sure, she might prescribe me antibiotics anyway, because she's treating me. Public health is treating the community. And antibiotic resistance is a public health problem. So when we look at this difference, what is best for one patient may not be best for the community. As the famous saying goes, the needs of the many outweigh the needs of the one. And I know I'm getting old, and a lot of you getting old compared to your age, and a lot of you may not recognize that quote. It was originally said by Spock in one of the original Star Trek movies. It's logical, right? The needs of the many outweigh the needs of the one. But if you're the one, or your kids the one, or your parents are the one, or your loved ones are the one, it gets personal. And you tend to think about yourself. You tend to be a little more selfish. This is a map that shows prescriptions per population density, so per 1,000 people in the population. And this came from a study that was released in 2016 in the prestigious Journal of the American Medical Association, that it is a prestigious journal, that looked at an estimate of how many prescriptions were unnecessary. And it found that in the US, about 30% of antibiotic prescriptions were unnecessary. The person was not likely to die, or it wasn't even bacterial to begin with. And we can see the highest rates here in the darkest colors. So Arkansas, Louisiana, Alabama and Mississippi, Tennessee, Kentucky, West Virginia, Nebraska. Next up, we have like South Carolina, Missouri, Iowa, Kansas, Oklahoma. A lot of this is the central part of the country. The West Coast has the lowest rate of antibiotic prescriptions. Why that is, I will leave that up to you guys to think about that. I have some ideas about why some of these areas have higher rates than others, but it's not a nice, neat little clean cut sort of concept here. Okay. The next category I'm going to talk about for overuse of antibiotics and problems with antibiotic resistance. And this is problems with antibiotic resistance. Not so much the overuse of antibiotics. And we're going to call this public sanitation. This is looking at places like schools, libraries, public transit, hospitals, doctor's offices. And you want to ask yourself, how often are these cleaned? And how well are they cleaned? Let's be honest, the people that clean public spaces are not well paid. I don't know about you, but if I'm not well paid, I don't know how rigorous a job I would do. Okay, I say that and I'm teaching at community college where I'm not real paid, and frankly, I work my ass off. But for most people, if you're cleaning, how good of a job, it has something to do with whether it's your space and how much you're paid. Let's be honest. So we have to think about this. If the surfaces are not well cleaned and the bacteria can survive on those surfaces, antibiotic resistance can spread between different bacteria, especially in places where people tend to be sick. People go to school sick. You know, when I was a kid, they got mad at you if you took too many sick days, and they gave awards for not being sick all year. Well, coming to school while you're sick, let's be honest. That's not good for public health. Public transit, I'm sure that's not cleaned well enough. Hospitals and doctor's offices, a lot of sick people come in, there's going to be a lot of bacteria. Now, antibiotic resistance, one of the big ways it can spread around the hospital is right here. If you go to your doctor or a hospital and the doctor comes in wearing a tie, yeah, that's not a good thing. These ties aren't usually cleaned very often. And so they can carry bacteria all around the hospital. Same thing with the shoes. Do you clean the bottom of your shoes very often? Most people don't. You can carry the bacteria around, including including antibiotic resistance bacteria from patient room to patient room on the bottom of the shoes. Now, how often should they be cleaned is a totally different question. Some places should be cleaned very frequently, but some degree of exposure to so-called germs is good for your immune system. We don't have a good way to measure how much exposure is good for you, but we know that if you are, especially if you grow up in an environment that is too clean, that can increase the risk of autoimmune diseases later in life. Autoimmune diseases are when your immune system attacks your cells. Okay. So like I said, we don't have a good grasp of how much exposure to germs is good for you. It's just not clear yet. There's also this idea of individual responsibility. And here are some of the problems with that. There are people who will demand antibiotics. And if you're a doctor and your patient says, I want antibiotics, or they're a mother and they go, my kid has an ear infection, give them antibiotics. If you say no, they may go to a different doctor and get the antibiotics anyway. Now, the vast majority of ear infections are viral. Antibiotics does nothing for the vast majority of ear infections in children. Giving antibiotics in those situations, you might feel better, you're doing something rather than just waiting for your immune system to do it. But it's increasing the likelihood that we're going to get more antibiotic resistance. We also know that up to 40% of people quit medication early. Now, some of these, you know, if you're getting a rash from taking an antibiotic, you do want to stop that antibiotic because that's a problem. But if you don't have weird symptoms like rashes, you need to take the full course, because what we saw on that video, we saw bacteria starting to evolve resistance to low levels. And once they develop low resistance to low levels of antibiotics, then they can have changes and resist higher levels. If you stop the antibiotics early, it's like exposing those bacteria to low levels of that antibiotic. They can develop resistance to the low levels, and then next time you get sick, they already have that first mutation that can lead to more antibiotic resistance. And people quit their medications. Most of them, they feel better. The drugs are working, I'm not sick anymore, I don't need it anymore. That doesn't mean you're completely cured of that particular infection. It just means you don't have symptoms. You don't have enough bacteria left in your system to trigger the symptoms you felt. Sometimes it's side effects. Like I said, rash, that's a bad side effect. Tummy troubles, that's kind of to be expected with most antibiotics. So that's kind of a problem. And of course, in lots of places in the world, you can purchase antibiotics without a prescription. And that's going to lead to a lot of problems. I also want to talk about agriculture. Around 1950, an experiment was done that showed that chickens given what's called a broad spectrum antibiotic, which means it tackles gram-positive and gram-negative, lots of different bacteria are affected by this. These chickens grew faster and bigger. Which is great for the farmers, right? The faster you grow the chickens, the less time and resources you have to put into them before you can sell them for food. And bigger, more meat, more money. Same thing happened in pigs. And so pharmaceutical companies started making specifically tetracycline available in nutritional supplements for agriculture. So they were literally putting tetracycline in the food. These were low doses. We already know low doses are not a good idea. So low doses strongly select for resistance. Another great advantage for agriculture, antibiotic use means you can pack animals in tighter quarters. And there we go. Crowd them in, give them more individuals in less space. Before the widespread use of antibiotics, animals were spread out further. They had more space because if you get a disease and the animals are packed closely together, it can spread through the whole flock or the whole barn full of pigs. And then you've lost your livelihood for the year. If there's more space between them, it takes longer for a disease to spread between individuals, and you might have a chance of catching it before it wipes you out for the year. These antibiotics also get in the waste of these animals. So their feces in particular, but their urine as well. And they can end up in nearby waterways, especially hog farms. So hog farms, hogs produce a lot of waste, and it's rather stinky waste. And the way hog farms have been dealing with the amount of waste that they produce is they put manure in massive lagoons. So there will be a big pond-looking structure, and that's where they put the pig waste. And during hurricanes or floods, that basically sewage gets washed into local waterways. Horse manure, cow manure can be composted in different ways that are less dangerous for local waterways, as long as it's composted. Same with chicken manure. Chicken manure can be composted pretty well. In 1971, the United Kingdom, England, and associated countries banned the use of antibiotics as growth factors. It was available to treat diseases, so that was great. Unfortunately, they could still be used prophylactically or as a preventative. So the use of antibiotics did not go down. There were still just as many antibiotics being used in agriculture. In the US, the FDA was looking to do the same thing. And a single representative stopped it. So representative, this is somebody in the House of Representatives, Jamie Whitten, was powerful enough that he threatened not to fund the FDA if they regulated antibiotics in agriculture. So it took, instead of doing this in the 70s, it took until 2012, the US finally regulated use of antibiotics in agriculture, which of course the usage actually went up after this. So it didn't do what we wanted it to do. One last tidbit in this topic of agricultural use of antibiotics. Recent studies have shown that antibiotics no longer promote increased growth in farm animals. It did in the 50s. It's not now. Why? Well, that's a good question. And we will come back to the why next time. Any questions on anything we covered today?
No
You're
questions
probably
about.
gonna.
Oh,
Sorry.
go for- no,
You can
go
speak
for it, go
first,
for it.
Will.
All right. I was just going to say the journal entry for April 16th. It's in the system, I think, as a test. So I go to click into it and it says there are no questions. You can't complete this.
Okay, I will fix that.
Okay. You're probably going to answer this in the next class, but in terms of antibiotics, would it be like if you were to consume like, chicken or pork or beef that had antibiotics in it, would that mean that your resistance to that said antibiotic would increase?
It could.
Yeah.
Okay. That's the short answer. Like you said, I'll get to that one next time in a little more detail. All right. In that case, I'll say goodbye and see you guys next time.
Thank you. Have a great day.
Thank you. Bye.
Thank you.
Thank you.
Bye.